Assalamu alaikum, guys. Good evening. So today we're going to discuss a little bit of more uh, in detail discussion about enzymes. In the last lecture, we discussed about the basic concepts, what enzymes are, how they function, a little bit of idea about different sort of competitive and non-competitive inhibitions, and the factors affecting the rate of enzyme activity. What we are going to do today, we are going to discuss that mechanism, that how enzyme actually work, what is the kinetic model of enzyme activity, how the inhibitors will affect the enzyme activity, how different inhibitors and their mode of action differ from one another, and how we can control that inhibition as well. There will be a bit of mathematical involvement in this presentation as well. Uh, there is a mathematical derivation that we're going to discuss in this. Definitely, I'll go through the derivation, but this is just for understanding purpose. Like it's not like that you have to learn the whole derivation for the exam, but rather it's more important to go through the derivation and understand what's going on in this and how the derivation can be used to understand the enzyme kinetics, enzyme inhibition, and the control models. So let's start our discussion. As we discussed last time, that there are different inhibition model for enzyme activity. And I told you last time that not all inhibitions are considered to be as bad. There are some inhibitions of the enzyme activity that can be used for therapeutic purpose as a drug. So we know that there are many drugs which are enzyme inhibitors, and some of the examples are right in front of you, like Lipitor, which is an HMG-CoA reductase enzyme inhibitor. Now, this particular enzyme is a liver enzyme, and this is important in the synthesis of the cholesterol. So you can see that this inhibitor is a drug that is Lipitor, which is going to stop HMG-CoA reductase, and thereby it stops the biosynthesis of the cholesterol. So you can easily figure out that this is for cholesterol lowering drug. Similarly, Vyriad and Emtreva, they are the inhibitors drug used for reverse transcriptase inhibitor. This is an enzyme which is doing the reverse transcription, and these drugs are the inhibitor of that, and they are antiretrovirus or antiviral drugs because they are used in the treatment of HIV. Then we have tequinavir. This drug or this inhibitor is a protease inhibitor, which stop the protease enzyme, and this is also one of the antiviral drug used. So we can conclude that inhibition has got different aspects. Sometimes this inhibition by a particular inhibitor could be, uh, you know, a factor that could result toward a disease production. While in some cases, we might have in these inhibitors being used as drugs for the treatment of certain diseases. So, how can we define enzyme kinetics? Enzyme kinetics is actually the study of the reaction rates, which are time dependent phenomena. Because as we mix an enzyme with a substrate, this is what we call the time zero. And then on providing the optimal conditions like temperature or pH, and other factors, the reaction will progress. And after a particular time, we have got the product formation in front of us. So this whole mechanism of how the enzyme interact with the substrate and what is the detailed process in terms of reaction rates, that is what we call enzyme kinetics. If you remember last time we discussed about a little bit about this kinetics in terms of energy of activation, and you have been told that enzymes actually work by lowering the energy of activation. And what is energy of activation? The minimum amount of energy required to transform a substrate into the intermediate state, from where then it's going to be transformed into product. Now, the rates of the reactions can be affected by many factors, and some of these factors we have had the discussion in the last lecture, like the substrate concentration, like the temperature. But there are few which are first time we are going to discussing here, like enzymes, catalyst, concentrations of certain other things besides substrate, and effectors molecule, which are actually not the direct part of the reaction, but somehow these are the molecules that can affect the enzyme reaction rate. So there could be different factors, there could be different things that can affect the rate of the reactions, and we'll see how it actually works under the light of enzyme kinetics. Now, the question here that why we study enzyme kinetics, what is the benefit of studying that? Now, there are few answers to this question. Number one, the enzyme kinetics is used for the quantitative description of biocatalysis. Quantitative description means how much quantity of a particular enzyme, how much quantity of a particular substrate is needed in order to carry out that particular reaction. That can be figured out, that can be determined if we know the proper enzyme kinetics of that particular reaction. Similarly, enzyme kinetics is also important to understand the mechanism of the catalysis, like how the enzyme and its substrate, you know, get in touch or, attach with one another. What is the mechanism? Like, for example, in the last lecture, we discussed about lock and key model, we discussed about induced fit model, and, all these kind of things, how an intermediate transition state is achieved, and then how that intermediate or activated state can be transformed into a final product. So the whole catalytic mechanism will be understood when we study the enzyme kinetics. Then also very important part of enzyme kinetics is to find out the role of and the effectiveness of inhibitors, because when we know the mechanism, when we know the enzyme kinetics, we can figure out what sort of effect can be produced by an inhibitor on the enzyme, because knowing the mechanism will give you an indication that how the inhibitors are working, what is the mode of action of the inhibitors for that particular enzyme. And of course, last but not the least, the enzyme kinetics is important to understand the regulation of the activity, because as we said earlier on that every enzyme catalyzed reaction, every biochemical reaction must have regulation. The reactions within the cell are not going to be in an uncontrolled manner. There should be some sort of mechanism by which we can regulate different biochemical reactions depending upon the need of a particular product. So in some cases, we need to enhance a particular reaction to get the final product. And in some cases, we might need to stop or slow down a particular reaction. So in order to understand how it is being done, how it is being regulated, we should know the enzyme kinetics. Now, there are a few general observations which I'm sure that you also might have an idea about. These general observations are part of enzyme kinetics. What are these observations? Number one, enzymes are able to exert their influence at a very low concentration. Now, why this observation is there? If you remember my last lecture, we discussed that enzymes are protein. They are big in the structure. And an enzyme can have multiple active sites. So let's say, for example, just giving you an idea, if there is one molecule of enzyme that may have 10, 15, or 20 active sites. And on those active sites, it can bind with the substrate. So one can say that one enzyme molecule is enough to bring about the catalysis of 15 or 20 molecules of the substrate into their final product. So that means the requirement of the enzyme in terms of their concentration is low. So very low concentration of enzymes are generally needed. That's what we say the enzyme is equals to nanomolar. That means very small concentration. This is one general observation. And I told you the reason for that. The second general observation is that the initial rate or the velocity is linear with enzyme. What does that mean? The initial rate or the velocity means once we mix the enzyme with the substrate, as soon as the reaction starts, it's linear with the enzyme concentration. That means if you increase the concentration of the enzyme, the initial rate or the velocity of the reaction will also increase. But make sure that we are talking about initial. That means after a particular time, that is not the case. So the initial rate or the velocity of a biochemical reaction is directly proportional with the concentration of the enzyme. This is our second general observation. The third observation is that the initial velocity or initial rate, what I said, increases with the substrate concentration. That means when you have increased amount of substrate, or in other words, when you increase the substrate concentration, the initial velocity will also increase. But that will be happening at low substrate concentration. What does that mean? For example, if you have got one enzyme molecule, let's say, and you have got, let's say, 20 active sites on that, and you provided a very low substrate concentration, for example, two molecules. So these two molecules will bind to the active site, but there are 18 more active sites still vacant. So now if you increase further substrate concentration, it will increase the initial velocity. Why? Because active sites are still available. That's what I want to say, that the initial velocity is directly proportional to the concentration of the substrate at a low substrate concentration. When the substrate concentration reaches I, that means there is a point where enzyme gets saturated and there are no more active sites available. And at that case, increasing the substrate concentration may not have an impact on the overall reaction velocity. So this is the third general observation. The fourth general observation is the initial velocity approaches a maximum at high substrate. When the reaction starts, as I said, we call it initial velocity. And gradually, the reaction velocity is going to increase until a point where it approaches the maximum point. That is what we call the maximum velocity or the Vmax. And that Vmax will be achieved at a high substrate concentration. So I think this point is related to the previous point. Like when you have low substrate concentration, you've got more active sites available. You keep going on increasing the substrate and they got attached with the enzyme. The reaction velocity increases up till a point where enzyme is fully saturated. And that's the point where you have the maximum velocity. So these are few of the general observations that we have had the idea when it comes to enzyme kinetics or the rate of the reaction. Now this is the picture, a graph of the same observations that we have discussed. Just to let you know, guys, that it's almost more than half of the syllabus that we have covered, but still you guys are not following the instruction, and still people are joining late in the class. I don't know for what reason, but I'm not going to add any one of them. So please do communicate to the class fellows that if the time is 5:30, you need to be there at 5:30. If you cannot for any genuine reason, fine, keep yourself away from the class and listen to the recording, because I don't want the distraction in the class again and again. But I don't know why people still do not follow a very basic rule of coming on time and keep coming in the class at this stage. Okay, so now going to continue that this particular graph is an enzyme kinetic graph, which is being between the product concentration versus the time. You can see on the y-axis you have product concentration, and on the x-axis you have the time. So you can see that there are different concentration levels which has been shown. For example, this one where the substrate concentration is 1 micromolar, and this is when the substrate concentration is zero point 0.2 micromolar. So obviously, here we have a lower substrate concentration, and here we have the higher substrate concentration. So with the lower substrate concentration, you can see the reaction starts, initial velocity increases a little bit, and then it became pretty much constant. That means the Vmax is achieved here, right? In this case, when you have the higher substrate concentration, as we said earlier on, there is a sharp increase as a linear way with the increase of the velocity and the substrate concentration. And ultimately, it reaches this point where you have got kind of Vmax achieved. Similarly, this is what we call the Km. Km is actually that concentration of the substrate at which half of the velocity is achieved. So you can see that this is the highest, this is the lowest, and this is the middle point. So that concentration, which is in this case zero point 0.5 micromolar, is the concentration of the substrate at which half of the velocity is achieved, and that particular is called Km. So you can see that this dotted line starting from these points is actually reflecting the initial velocity. So you can see initial velocity is increasing there, it's increasing there, it's increasing there. So that means in generally, initial velocity is increasing with the increase of the substrate concentration. So that is the outcome, that the initial velocity increases with substrate concentration. But as I said earlier, at low substrate concentration. Once it is at the high substrate concentration, then there will be saturation and there will be no increase in the velocity. Now, this is the diagram, a particular graph which is showing the enzyme kinetic model of an enzyme. And look at this graph, that was between the product and the time. But now the graph is between the velocity and the substrate concentration. So what we have seen, it's very simple to understand, that with the increase of the substrate concentration, there is a sharp increase in the initial velocity. And it carries on when you increase more substrate, you get more velocity, and finally it reaches to this point where you can see it's becoming pretty much flat, and this is what we call the Vmax. Vmax, as I said earlier, is the maximum velocity. So the initial velocity approached a maximum velocity at a higher substrate concentration. And we can see from this graph very clearly that at this point, if you increase more substrate concentration, it's not going to impact the reaction velocity. Why? Because as I said earlier, the enzyme gets saturated. So here the initial velocity, which is called the V naught, was measured in micromole per minute, while the the substrate concentration was in millimole. And as I said in the previous slide, the half of the Vmax is being called as the Km. So this is the initial velocity. Here you have the Vmax, and half of that is the concentration of the substrate at this point will be called as Km. Km is a constant. Different enzymes have different Km values depending upon their structure and the binding of their substrate. So this is a typical enzyme kinetic graph of a model where it shows that initial velocity increases with the increase of the substrate concentration till it reaches the point where the maximum velocity is achieved. And this is where we call the saturation of the enzyme takes place. And half of the Vmax will be called as the Km value. So what we conclude is that the initial velocity increases with substrate concentration at low substrate, which we have already shown in the last slide. And velocity can be determined as D is the change of the concentration of the product divided by change in time. So definitely with the time change and with the change of the product concentration, we are determining the velocity of the reaction. So I'm sure at this stage, these basic concepts are clear. Now, there are certain mathematical equations, as I said earlier on in the start of the presentation, that there will be a couple of mathematical models to describe enzyme kinetics. And I told you earlier on that why we are interested in enzyme kinetics, because there are a couple of applications and uses for us, and that's why we are interested to know the enzyme kinetics. And in that way we develop different mathematical models to find out the enzyme kinetics of different enzymes. And with these mathematical models we are able to find out some useful information about how the enzyme is working, how its binding is affected, how the inhibitors are playing their role, and how the regulation of a metabolic reaction can be done. So all equations, all mathematical models of describing enzyme kinetics, they start with a model. We call it a mechanistic model. Then after designing a mechanistic model, like how the mechanism takes place actually, we identify constraint and assumptions, like what are the constraints that can affect this enzyme reaction, and we'll do some assumptions, which is the part of mathematical model. Then we'll do the algebra, the real mathematics, and then we calculate the velocity. So all equations, all mathematical models of enzyme kinetics, it starts with a first with a model, that how the reaction is going to take place, what are the constraints that can affect the reaction, and we'll do some assumptions to start up with. We do the mathematics and we solve the velocity, which I already mentioned, that velocity is the change in the product concentration divided by change in the time. Okay, let's start with the first and the most common enzyme kinetic model we use in biochemistry, and that's called Michaelis-Menten kinetics. These were actually two scientists, Michaelis and Menten, who derived this mathematical model of enzyme activity, and based on that, it's being called as Michaelis-Menten kinetics. Now, there are a few common things you need to understand before we go in the detail. This is the model which is showing that we have this enzyme and we have the substrate. So when enzyme substrate mixed, it results in the formation of an intermediate, which is called enzyme substrate complex. And I'm sure that you guys remember, you have been told about this when we discussed the induced fit model. So this is what we call the transition state, or the intermediate state where the enzyme substrate complex is formed. There is a constant for this reaction. Every chemical reaction, has got a constant. So the constant for this chemical reaction is K1. Now look at the reaction, that this is a reversible reaction. That means at this stage, this enzyme substrate complex can be converted back to the free enzyme and substrate. So that's why it's shown here with the reversible arrow. And the constant for that reversible reaction is, let's say, K minus 1. So one understanding is that when enzyme is attached with the substrate, it results in the formation of an enzyme substrate complex. And second important point is that this intermediate can be transformed back to enzyme and substrate as well. So at this stage, we can say the reaction is reversible. The next step is that this enzyme substrate complex will be transformed finally into an enzyme that is a free enzyme and the final product. The constant for this stage is called K2. So a simple enzyme mechanism goes by one reactant, one intermediate, and one product. This is what I said earlier on, a mechanistic model. So the mechanistic model of the Michaelis-Menten is this one. We have one reactant, one intermediate, one product. Enzyme is the common because enzyme starts and then we got the enzyme back here once the reaction is complete. So remember about these two steps, that enzyme substrate complex formation is a reversible reaction, and then you have got different constants for this and this. Okay, so in order to understand the Michaelis-Menten kinetics, and again the same equations are there in front of you. I'll go through in detail with that Michaelis-Menten kinetics step by step. But as I said that, I really want you guys to understand how that model works. Obviously, these equations and the formula are important, but it's of no use to just learn the whole equation without understanding what's going on. So I would recommend to go through step by step and you see how the transformation takes place. So the first step is the enzyme in the substrate, they bind reversibly and quickly, forming a non-covalent enzyme substrate complex. I think first point is pretty much clear. I already explained. Couple of things I want to add on. Number one, enzyme substrate complex is a non-covalent complex. That means the enzyme and substrate, they are not bound with a covalent bond. That's why it's reversible. Second step, the enzyme substrate complex will undergo now a chemical transformation. And as a result of that chemical transformation, it dissociates to give the final product P, and the enzyme is free. So two steps are clear, and I think these two equations are pretty much the same that I explained. The third step is velocity V is equals to K2 and the concentration of enzyme substrate complex. Now, why this is the case? Why velocity? If I remove this K2, let's say, you know that this velocity will be directly proportional to enzyme substrate complex concentration. Why the velocity is directly proportional to enzyme substrate complex formation? The reason is that this step is reversible. So activity or the velocity is not here directly corresponding to this concentration, because if it transforms to this, it's a reversible product. It can go back. But once it reaches here in the second step, which is, the coefficient is K2, the actual reaction velocity is depending on this concentration of enzyme substrate complex. That's why velocity is equal to K2ES. Now, next step, many enzymatic reactions follow Michaelis-Menten kinetics. This statement is itself reflecting that not all enzyme reactions follow this. And as I said earlier on, that there are many other mathematical models to explain the enzyme kinetics as well. But many enzyme reactions within our biological system follow the Michaelis-Menten kinetics, even though enzymes mechanisms are always more complicated than the model itself. For the real enzymatic reactions, we use Kcat instead of K2. So remember, Kcat, cat stands for catalysis. So Kcat is actually K2 according to this, equation that you know, and you can always remember wherever you see Kcat, that means you're talking about K2. And obviously, this is the real enzymatic reaction because here you are getting the product. K1 and K minus 1 are not actually directly affecting the product formation. Okay, so what is the enzyme substrate complex? You got the idea. The enzyme will bind non-covalently with the substrate to form a complex, a non-covalent complex called enzyme substrate complex. The ES complex is also known as Michaelis complex. A Michaelis complex is stabilized by molecular interactions which are non-covalent. Like it could be hydrophilic, 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 or hydrophobic, hydrophobic, or different other kind of non-covalent interactions like hydrogen bonding, disulfide linkages, and etc. etc. But that's not a covalent interaction. That's the most important thing. The Michaelis complex form quickly and also dissociate quickly. So I think this point is pretty much clear that formation and reformation of the free enzyme and substrate can occur. That's why we are saying that this is a reversible reaction. So the kcat and the reaction velocity. We already determined that the constant which is going to affect the reaction velocity is actually kcat, because we are talking about this particular portion where enzyme substrate complex is transformed into enzyme and product, because that's what the intermediate state is. So velocity can be defined as a small change in product concentration with reference to small change in time. In other case, we can say that this velocity is equals to k cat es according to this equation here, this one, right? So we have just substituted this value. In place of this, we substituted this, and then we said V max will be the maximum velocity will be equivalent to K cat into concentration of enzyme. Why we have changed here with the enzyme? We remember that one enzyme molecule can have more than one active site to accommodate the substrate. So when we talk about the maximum velocity, actually, not I'm talking about this velocity, the maximum velocity will be achieved when you have the enzyme concentration affecting the overall reaction speed. So that's why we have substituted E naught concentration here, and we say that Vmax is equals to K cat E naught, and just rephrasing that, the K cat will be equivalent, K cat will be equivalent to Vmax divided by E naught, just shuffling this. So the enzyme is either free, that is in this form, or bound, that is this form. The unbound is equal to the enzyme substrate complex plus the free enzyme. Like for example, you have got 10 moles of enzyme and 5 moles of enzymes attached with the substrate. So how will you calculate the total enzyme? The total enzyme will be the enzyme that is bound with the substrate plus whatever enzyme left behind. At sufficiently high substrate concentration, we know that all the enzyme is tied up. That means at a high substrate concentration, all the enzyme has been transformed into enzyme substrate complex. So at this stage, you can say that E naught is approximately equivalent to the total enzyme substrate complex. And that is according to the chemistry's Le-Chatelier's principle. At high substrate concentration, the enzyme is working at a full capacity. So that's why the velocity will be called as Vmax. The full capacity velocity is determined only by K cat and the total enzyme concentration according to these equations. So kcat can be termed as a turnover number. That means the number of the moles of the substrate produced per time or per enzyme active site. So finally, what we concluded that the kcat is the turnover number, and the turnover number can be defined as the number of moles of substrate produced per time per enzyme active site. So this is how the kcat is defined. So for an enzyme, it is possible, or it's pretty easy to determine the kcat, because that's what is affecting the enzyme overall reaction. To understand and compare enzymes, we need to know how well the enzyme binds with the substrate. That is what happens in the first part of the reaction. Kcat does not tell us anything about how well the enzyme binds to the substrate. Now I'm going back to my point. I said that enzyme velocity and enzyme kinetics is mainly dependent on the kcat. Why? Because the enzyme velocity is actually being determined when the enzyme substrate complex is dissociated into enzyme and product. But what I'm saying that the kcat is not telling us that how the substrate bind with the enzyme. That has been taken before the kcat. So in this case, we have to go back and see what this K1 and K minus 1 was, which we got the KD and KM here, and we have to understand the importance of that as well. That means, in order to understand a couple of important things, we cannot ignore the first part. First part means the formation of enzyme substrate complex. We also want to look at this one in order to understand a couple of important aspects of enzyme kinetics. So, as I said, now we have the model, we have different influencers, and now we go towards assumptions to deduce the mathematical form. The assumptions we have is the K1 and K2 are far greater than K2. Sorry, K1 and K minus 1 are greater than K2. That means the reaction constant of enzyme substrate complex formation and enzyme substrate dissociation is greater than the rate constant of enzyme product formation. That means the first step is fast as compared to the second step. Or in other words, the first step is always at equilibrium. So this is our assumption number one. That means the formation of enzyme substrate complex is a quick step and it's a rate determining step. And then from enzyme substrate complex to enzyme and product is a slow step. So always the first step or the slow step is being referred to as the rate determining step, because if that doesn't happen, then there will be no product formation. Second assumption is that change in the enzyme substrate concentration with reference to change in the time is approximately zero. That is what I call the equilibrium, and that is the system is at steady state. So that means the change of the enzyme substrate concentration with reference to the change of the time is pretty much zero. That means as soon as enzyme substrate complex is formed, it's being transformed into enzyme and product, or back to enzyme and substrate. So based on these assumptions, we can say that the change in the concentration of enzyme substrate complex reference to time is actually the rate of the formation of enzyme substrate complex, or the rate of the breakdown, because that's how we are saying that it is at the equilibrium stage. The third assumption is that there is a single reaction dissociation step. That means actual dissociation takes place at the second step, where the enzyme substrate complex is transformed into enzyme and product, and that is K2, or we can call it Kcat. So overall, in total, we can say that it is the combination of substrate concentration plus the concentration of enzyme substrate is approximately equivalent to substrate concentration. What does that mean? That means whatever amount of substrate we have provided has already bound with the enzyme to make an enzyme substrate complex. And that is why we say it's approximately equivalent to the total substrate concentration. The fifth assumption, there is no back reaction of product to enzyme substrate. And I think this assumption is pretty much clear, that you can see that this is not a reversible reaction. So there is no back reaction of P to ES. Once the P is there, it cannot go back. That means the concentration of P at this stage is approximately zero. I'm talking about this stage, right? This assumption allows us to ignore K2. When we say the concentration of P is zero, that means this reaction is not going on. And when this reaction is not going on, that means K2 can be ignored. So now our entire dependence is on this first step we are talking about, right? So we measure initial velocity when the concentration of P is zero. Initial velocity obviously is the velocity that refers to the beginning of the reaction. So once you mix the enzyme and substrate and just the reaction is started at time zero, for example, we expect that there is no product because reaction has just started. That's why we are considering the product as zero at this stage. So we have 5 assumptions overall. I'll go through them quickly. And what I'm going to do is, like, I'm going to stop my today's lecture at this slide so that you will go through it. So what are the assumptions? Number one, the the constant of the reactions of enzyme substrate formation and enzyme substrate dissociation is greater than K2. That means the first step of the reaction, I'm talking about this one. The first step of the reaction is fast and is always at equilibrium. That means the way it is being formed as ES will be the way it is being converted back to this. That's why it's at equilibrium. We say that change of the ES with change of time is zero. That is, the system is at steady state. So we can say that this one can be either called as the rate of formation of ES, or you called it a rate of breakdown of the ES, because that's what the equilibrium is. The third is there is a single reaction dissociation step, which is this one. Dissociation is actually going on here. Although dissociation is this one as well, but we are saying that that is at equilibrium. So we consider dissociation is actually taking place here, where the enzyme substrate complex is converted to enzyme end product. And that reaction is catalyzed, and the constant is K2. So we called it Kcat. The fourth assumption is that the total substrate concentration is actually the sum of substrate plus enzyme substrate, and that is roughly equivalent to substrate concentration. Why? Because if we have, let's say, one enzyme and we have got 10 substrate molecule, and the enzyme has got 10 active sites, all of the sites have been occupied. So one can say that the total substrate is roughly the sum of the substrate plus the enzyme substrate complex, keeping in mind that that is in equilibrium. If it's going in the front direction, it's going in the back direction as well. So overall concentration would remain the same. And the fifth assumption, which is very, very important, that there is no back reaction of P to ES. We all know. That means P is zero at this stage. This assumption will allow us to ignore the K2. Why? Because we measure initial velocities. And initial velocity means once the reaction has just started. And considering at this point, the concentration of the product would be zero. So these are the five assumptions. We keep it in mind. And we'll consider these five assumptions to then do the algebra and mathematical part of this Michaelis-Menten kinetics. So I'm going to stop the lecture here, and the forum will be open for the question. If you have any question, you can ask the question at this step, or if you wanted to ask later on, you are more than welcome. So if you have any question, just raise your hand. Okay, so if there are no question, I'm going to stop the presentation here, and I'll share the slides and as well as the recording with you. Thank you very much, and for the office.